With most cell types, you can optimize your transfection even further to get better results. A major reason for optimizing transfection lies in the current cell biology research trends. So far, most researchers have been using well-established cell lines that are relatively easier to transfect. However, more and more research is trending towards harder to transfect cell types, such as primary cells and embryonic stem cell products. Furthermore, we know that not every cell type behaves similarly with every transfection reagent. That makes it more imperative to optimize your transfection conditions to get that additional transfection output from your experiments. Improving transfection performance is fortunately not very hard to do. The answer lies in the simple transfection workflow if you just look at it sequentially. You begin with looking at your serum-free media for complex formation, the nucleic acid that has to be transfected, the complex formation time, cell culture conditions such as confluency or suspension cell density, and in the end, the harvesting time for your assay. The serum-free media that you use for your transfection complex formation is one of the most critical factors in transfection. The reason serum-free media is recommended for preparing pretty much all the transfection complexes across different transfection reagents is because serum is known to contain nucleases that are likely going to chew up your nucleic acid substrates even before it sees a transfection reagent. So it is an absolute no-no to use serum-containing media for complex formation. Another major factor that could disturb transfection is antibiotics. A lot of antibiotics, such as kanamycin, can be highly charged and can disrupt transfection complex formation. On the same theme, other highly charged compounds, for example, polyanions such as heparin sulfate, can disrupt transfection complex formation. We also know that in certain applications, for example, therapeutic protein production, where suspension cells are cultured in large bioreactors and are maintained in the presence of surfactants such as pluronic F68. This compound can significantly obliterate transfection efficiencies. Therefore, all of these categories of compounds or highly charged molecules should be completely avoided at the step of transfection complex formation. If needed, some of these compounds can be added later on during your transfection incubation. To further emphasize how much serum-free media can affect transfection, here is some data with GFP transfection of 3D3 cells with complexes made using various different serum-free media. As you can see here, serum-free media such as OptimM, DMEM, and RPMI 1640 afford decent levels of transfection efficiencies in this case. However, I would like to particularly note that certain media such as 293-SFM2, the HiQ series, the XL293 media, and the HiQ CDM CHO media completely obliterate transfection. One major cause for this inhibition of transfection could be that these media are known to be used for suspension cell culture, in which case they might have surfactants like prolonic F68 that are completely inhibiting transfection in this case. In specialized applications such as large-scale protein production, the overall transfection and therefore the protein yields can also be affected by the complete media that the cells are grown in. This is because in these applications, the final cell density is very, very important. Here is some data shown with our Transit Pro transfection kit in which an antibody-expressing plasmid is transfected into CHO suspension cells that have been adapted to a variety of different media. As you can see here, you get maximum expression in the case of CHO cells that are adapted to the freestyle CHO expression media, followed by the other media types. So in such cases where the final cell densities might be very important, it is highly critical to optimize even the complete media choice. The next critical parameter in the transfection workflow is the nucleic acid that has to be transfected. The most important attribute of the transfected nucleic acid is the level of its purity. For example, in the case of DNA transfection, it is critical that contaminants such as protein, carbohydrate, and lipids should not be present in the DNA prep as they may affect transfection efficiency. Most maxi prep kits that we use for DNA preparation in the labs do account for this purity level. We should also make sure that the ideal plasmid preparation exhibits an absorbance ratio of 260 by 280 of greater than 1.8 and ideally lower than 2. Like I mentioned, most maxi-prep kits allow us to get relatively pure levels of DNA. However, the biggest culprit when it comes to transfection is the endotoxin contamination of the DNA prep itself. This endotoxin is carried over from the E. coli cells because it is a part of the outer membrane of gram-negative E. coli. The endotoxin molecules are known to be very, very toxic to mammalian and insect cells. Here is some data illustrating the toxicity of endotoxins with respect to transfection. As you can see here with the commercial plasmid in which the endotoxin levels have been removed to very, very low amounts, you get good amounts of luciferase expression. However, with the crude plasmid that has exceedingly high levels of toxin, the transfection is completely inhibited. Now, when you take this crude plasmid and purify it and remove the endotoxin with 3 rounds of treatment with our Miraclean endotoxin removal kit, the levels of expression of luciferase are retained back to the same levels as found with the commercially purified plasmid. Therefore, it is very, very important to remove all traces of endotoxins from your plasmid preps for the best transfection. This is particularly more critical in sensitive cell types, for example, cell types of immunological lineage. The features of the nucleic acid that has to be transfected also plays a critical role in transfection. For routinely transfected nucleic acids, such as plasmid DNA, we all use common parameters such as CMV promoters, etc., that take care of maximal expression. However, in cases of substrates such as mRNA that are particularly sensitive, it is very important to consider the various features of the mRNA for maximizing transfection. mRNA can be easily produced in labs using an in vitro transcription reaction. However, the mRNA produced using in vitro transcription does not have the naturally occurring 5' cap structure, nor does it have the polyadenylated tail that helps with the mRNA stability and processing. There are a lot of commercially available kits now that let you add a cap analog on the 5' end or an iris that substitutes for natural capping. And also, polyadenylated tails can be added by using tailing kits that contain poly-A polymerase. Here is an example of how mRNA tailing can significantly improve expression. This experiment shows the expression of a luciferase transcript that is tailed and also another version that is non-tailed in raw 264.7 cells. The different colored bars represents the different times of harvest. And as you can clearly see here, at earlier time points such as 14 hours or even 24 hours, there is much better expression of the luciferase transcript when it is tailed versus non-tailed. So these small features in the nucleic acid can contribute to a great extent in maximizing your experimental results. Needless to say, the nucleic acid dosage also matters. We have seen routinely that doubling the dosage of DNA in harder to transfect cell types such as Jurkat cells or raw 264.7 actually improves the levels of expression quite a lot. Here is an example of how DNA dosage increases expression. In the red bars here is the ratio of 3 microliter reagent to 1 microgram DNA for 3 different cell types, COS7, CHOK1, and HEK293s. The gray bars represent the same ratio of the reagent to nucleic acid, but here the amount of nucleic acid is 3-fold lower. almost threefold lower, 0.4 microgram DNA. As you can see here, there is a corresponding decrease in the level of expression with the lower dosage of DNA. This dosage theme continues through other nucleic acid substrates such as mRNA. Here's an example of DC 2.4 cells that have been transfected with increasing dosage of a GFP mRNA from 0.5 micrograms to 2.5 micrograms. And as you can see here, not only is a larger percentage of cells expressing GFP with the higher dosage of the mRNA, they're also brighter. The same holds true for siRNA optimization. In case of siRNA, it's even more critical because higher amounts of siRNA can lead to non-desired off-target effects. As shown in the chart here, you can reduce the amount of siRNA to as low as 1 nanomolar to get effective knockdown. This also helps with any fears about off-target effects affecting your experiments. There are a few specialized applications that are affected with nucleic acid dosage. One such application is if you're overexpressing toxic proteins. The problem here lies in the fact that the overexpression of the toxic protein itself is going to kill the cells. In this case, a neat trick that you can employ is actually lower the plasmid dosage for the toxic protein. So for example, if you were transfecting 1 microgram of the toxic protein encoding DNA, you can lower it down to 0.1 microgram, that is 10-fold less, and add remaining non-competing cloning vectors such as a PUC18 or a 19 vector. Here is some data showing the expression of luciferase from a co-transfection of PUC18 and PCI-LUC. As you can see here, as you increase the amount of the non-completing cloning vector, the amount of luciferase expression decreases. In case of toxic proteins, this would be deliberate but would allow your experiments to succeed. The same holds for eGFP expression in this experiment, where you titrate down the amount of GFP to 0.25 micrograms from 1. And you can still see some GFP expression without having to hamper your experimental results. There are also different co-transfection scenarios that are routinely employed in the labs. One very popularly used co-transfection experiment is actually virus production. In this case, researchers typically take anywhere between 3 to 17 or even more viral plasmids and transfect cells together with them. Following transfection, the viable virus is produced and released outside of these cells in the supernatant. In most cases, people co-transfect multiple plasmid DNAs, but they can also co-transfect other nucleic acids such as multiple siRNAs, oligos, or mRNAs. It is very critical to always pre-mix multiple nucleic acid to prevent preferential complexation with the transfection reagent. If you have a mixture of 3 plasmids, always pre-mix them to avoid complexing one plasmid better than the other. Also, depending on your application, it is critical to choose the molar ratio of the multiple plasmids carefully. In cases of some reporter systems, only a very little amount of the reporter plasmid is added to the transfection mixture, whereas cases where you want to get equivalent expression from 2 plasmids, you would want to ideally add an equimolar ratio. There are also co-transfection scenarios where 2 different nucleic acids need to be co-transfected. Some examples are plasmid DNA and siRNA co-transfection, plasmid DNA oligo, or plasmid DNA and mRNA co-transfection. Of these, plasmid DNA and siRNA co-transfections are actually done quite routinely. One of our reagents, the Transit TKO reagent, works very well for this application. However, there are some points to consider when you're transfecting different nucleic acids. You should ideally form the complexes with each respective nucleic acid separately, again to avoid preferential complexation of one nucleic acid over the other. You can form these complexes separately and then just simply mix the complexes before adding to the cells. The amount of time that you let the complex form is also very critical in the transfection workflow and can be optimized in some cases. Most transfection protocols call for an incubation time of 15 to 30 minutes for transfection complex formation. During this time, the transfection reagent complexes effectively with the nucleic acid and also allows the right size of complex to form. The problem with letting it go too little is that you don't have the right size of the transfection complexes formed, and the problems with letting it exceed too long is that the transfection complexes become too big and are not able to be engulfed by the cell anymore. Here is an example of transfection complex formation time with plasmid DNA in CHOK1 cells. These cells have been transfected with a plasmid that expresses luciferase and is complexed with the Trans-LT1 reagent. As you can see here, anywhere between 15 to almost 50 minutes is a big window for the plasmid complex formation. Sometimes, though, we can see a precipitous decrease after 30 to 40 minutes, and generally we do not advise going further than 30 minutes of incubation for transfection complexes with DNA. This transfection complex formation time is even more critical with sensitive substrates such as mRNA. As you can see here in this chart, typically anywhere between only 5 to 10 minutes is required for effective transfection complex formation, in this case with CHOK1 cells that have been transfected using a luciferase mRNA. We do not advise going any further than 10 minutes in case of mRNA expression. Cell culture conditions such as cell confluency and density also play a very important role in transfection. In case of adherent cells, the overall cell confluency can have a huge impact on transfection. Here is an image of COS7 cells showing different levels of confluency from 25% to 90%. Cells that are very low confluency, for example 25%, generally are not very good in terms of overall cell health and therefore do not fare well with transfection. A typically good range for transfection is 50 to 70% cell confluency. On the other hand, very high cell confluency, such as 90%, lead to contact inhibition of the cells and prevention of cell division. In this case, the plasmid DNA is unable to be uptaken by the nucleus because of the dissolution of nuclear lamina that occurs in dividing cells, and that really hampers transfection efficiencies. To illustrate this point, here is a chart of 3D tissue cells that have been transfected with a luciferase encoding plasmid at 75% confluency versus 100% confluency. As you can see here, because there is active cell division at 75% confluency, there is higher amounts of luciferase expression. However, when the cells are 100% confluent, they are really contact inhibited, and there is no room for cell division whatsoever. This further translates in terms of lower luciferase expression. In case of applications where suspension cells are cultured, the overall suspension cell density also impacts transfection, much like adherent cell confluency. The applications in which this is affected are large-scale protein production applications, in which the optimal cell density typically tends to be anywhere between 0.5 to 1 million cells per mil, as shown in this chart below, of CHO suspension cells that are transfected with an EGFP plasmid. At lower cell densities, there is not enough cell mass, whereas at higher cell densities, there are again probably exhaustion of cell resources, which prevents the cells to actually transfect very well. Another very neglected issue related to cell culture is mycoplasma contamination. This can be very hard to detect because your cells look just the same as routinely cultured cells. They do not show any visible signs of contamination. That is not to say, though, that mycoplasma contamination does have a huge impact on transfection. It can lead to highly altered transfection results. In some cases, it leads to a severe decrease in transfection performance, whereas in some cases, you might get unrealistically high transfection rates. With a lot of PCR kits available now to detect mycoplasma, it has become easy to actually perform mycoplasma contamination tests in the lab. Our advice, if you suspect mycoplasma contamination, is to completely discard contaminated cultures. Or if you do not have that option with precious samples, you can always treat with antibiotics that are recommended for mycoplasma elimination. The last parameter in the transfection workflow that can be optimized is the harvesting time for your experiment. The harvesting times for different experiments are determined by the experimental goals themselves. For example, typical harvesting times for gene overexpression studies can range from anywhere between 24 to 72 hours depending upon the protein that is to be expressed. For gene knockdown, it is usually recommended to wait slightly longer, from 48 to 72 hours, to make sure that any residual protein expression that could contribute to misinterpretation of knockdown results is completely taken care of. For mRNA translation, which is susceptible to stability issues, earlier time points are desirable, for example, anywhere between 4 to 24 hours. Again, it might also depend on the mRNA itself. For specialized applications such as protein production, a longer incubation time is recommended, anywhere between 2 to 14 days, to increase the cellular biomass and therefore the associated protein yield. Here is an example of a human antibody that is produced using our Transit Co-transfection kit and also competing reagents such as linear PEI and Freestyle Max. As you can see here, in all three cases, there is an accruing of the antibody titers with increasing incubation time. So in this case, it is highly beneficial to actually let the harvesting times to be much longer. The harvesting times are also affected by issues related to nucleic acid turnover. Here is an example of Firefly luciferase mRNA that is delivered to NIH-3 cells using our Transit mRNA kit. As you can see here, possibly related to the issues of mRNA stability and turnover, earlier harvesting time points, such as a 12-hour time point post-transfection, is ideal for harvesting in this case. Before and after this time point, there is a rapid decrease in the level of expression of luciferase. This concludes our transfection optimization section of today's one-on-one talk. However, I would like to emphasize one last point when you're comparing transfection performance between different transfection reagents. Transfection results can vary quite a lot day to day based on the parameters that we discussed just in the previous slides. Therefore, it is very, very important that if you're doing any comparison of transfection reagents, that you perform the reagent comparison on the same day with the exact same batch of cells. It is also very important to follow reagent-specific protocols with each transfection reagent. Here is some data exemplifying this. In the first chart, you can see that reagent A outperforms reagent B if you compare day 1 transfections against day 2 transfections. However, once you have data from both day 1 and day 2, you can see that it's simply a matter of cell health in this case. It just turns out that the cells were transfecting much better on day 1. But they were transfecting much better with reagent B. So once you have the complete picture, you can see that in this case, reagent B outperforms reagent A. However, if you were comparing day 1 to day 2, that would not be the case. You can also learn more on our website, www.thetransfectionexperts.com. We have a technical resources section that you can find useful for gaining more information about transfection. We have tips from the bench there to help with particular experiment types such as DNA transfection or siRNA transfection. We also have a comprehensive transfection database called the Reagent Agent, which gives you recommendations for your particular cell type and nucleic acid combinations. You can also speak with the transfection experts directly by either calling us or emailing us at our tech support address. Thank you so much for watching and listening to this tutorial. If you have any feedback to give to us or any questions, please do contact us.